Hi everyone, my name is Ashlyn Sconiers, and today I'm presenting my formal paper over the impact of early nutrition on infant microbiome. I chose this topic because of my own journey with food allergies and striving to heal my gut. I also feel strongly about the benefits of breastfeeding and wanted to see if breastfeeding holds any significance in the development of a healthy gut microbiome in early life. I feel this also could be used as another topic of support for mothers in their decision to breastfeed over formula. So let's talk about the human microbiome. The human microbiome is a complex and dynamic environment and is comprised of at least 500 different species of microbes. The microbiome is a term used to describe all of the microbes of our body, including bacteria and fungi, which live in the intestinal tract of all humans. The clear majority of the microbiome lives within our gut, so this makes it one of the largest immune organs. For this reason, the microbiome plays a key role in the subsequent health of the host. The critical period of gut colonization, or the early procurement of bacteria, occurs within the first few weeks after birth and is influenced by a variety of internal and external factors. External factors such as the mode of delivery and antibiotic use exert an influence on the infant microbiome and has been shown to increase the risk of autoimmune and inflammatory conditions. You can see why the composition and diversity of infant gut microbiome plays a key role in the establishment of health. Infant gut microbiome development appears to be influenced by infant nutrition through breast milk or formula, and the diversity of colonization varies depending on the nutritional ingredient of these two. Therefore, I feel this topic holds important implications in health promotion, prevention, and the treatment of childhood diseases. However, I found the knowledge base on this topic of infant nutrition and gut microbiome is still rudimentary, and therefore the relationship between early nutrition and the development of the infant gut microbiome is a topic that warrants further examination. So the purpose of my paper was to examine the relationship, if any, between early nutrition, which is breast milk or formula, and the development of infant gut microbiome. Before birth, the neonatal gut is considered sterile and coated with amniotic fluid. At this point, the gut has yet to begin acquiring the variety of different species of bacteria that will make up the microbiome that the individual hosts throughout the lifespan. The diversity and composition of microbiome begins with the mode of birth, so vaginal birth or C-section, and is rapidly colonized within a few hours after birth upon exposure to the immediate environment, which can include the equipment, the air, other infants, and nursing staff that the infant is exposed to. There are complex mechanisms that contribute to the development and maintenance of the gut. These mechanisms include genetic components, environmental factors, inherent physiological functions, and dietary intake influences. When examining microbial colonization, intrinsic factors must be considered, such as neonatal genetics, gestational age, intestinal pH, and bacteria competition. Also, the extrinsic factors such as maternal health status, geographic area, mode of delivery, hygiene measures, and antibiotic use also have implications in the diversity and maintenance of the infant intestinal microbiome. I like this diagram. You can see it shows the development of the microbiome from within the mother's womb and through infancy into the toddler years. You can see the mother's own microbiome has an impact, vaginal infections, periodontitis, and then with the infant, things like antibiotics, breastfeeding, and environment that I had mentioned. So let's take a look at infant nutrition in the microbiome from what we know. Infant diet has been determined to have a direct influence on microbiome colonization by providing the nutrients needed for the propagation and function of bacteria. These two methods of feeding examined in my paper are exclusively breastfed and formula-fed infants, Both breast milk and formula comprise the same primary nutrients, which include carbs, fats, protein, amino acids, vitamins, minerals, and enzymes. Although formula is made to mimic breast milk, formula cannot. It lacks the immune factors, living cells, and bifidogenic nutrients breast milk offers that synergistically promote growth of good bacteria over pathogenic bacteria, making breast milk the hypothesized superior source of nutrition in the healthy colonization of the infant's gut. So let's compare the two, breast milk versus formula. Breast milk is unique due to its complex nutrient composition that can adapt to satisfy the infant's nutritional and immunological needs. Protein composition in breast milk includes casein and whey, and so does formula. The primary carbohydrates of breast milk are lactose and oligosaccharides, which are substrates for the proliferation of bifidobacteria, which is a beneficial bacteria. Breast milk contains immune properties due to its antimicrobial factors, immunoglobulins, cytokines, glycoproteins, lactoferrin, oligosaccharides, and lysozyme that can inactivate pathogenic bacteria individually or together. Standard infant formula is derived from cow's milk or soy milk. The typical cow's milk-based infant formula has high protein content composed of casein and whey, along with higher phosphate content. Supplemental ingredients are added to better replicate breast milk and can vary based on the type of formula. Added nutrients include vitamins such as iron, sodium, potassium, calcium, phosphorus, enzymes, amino acids, and even probiotics. Formulas can differ in source of fat content, but may include MCT oil, soybean oil, or coconut oil. Carbohydrate content includes lactose and a source of glucose, such as corn syrup or brown rice syrup. So how do these nutritional sources compare when we look at the microbiome? There are several differences in the microbiota of infant stool depending on the feeding type. When compared to breastfed infants, formula-fed infants have demonstrated a more stable and variable population of microbes. Differences in the nutritional composition of breast milk and formula hold an important implication towards supporting or disrupting a healthy infant microbiome. So when examining the breastfed infant microbiome, they showed to have a more stable and homogeneous microbiome. Breast milk's properties exhibit a direct role in bifidobacterium abundance due to its low casein, high whey, low phosphate, high lactose, low buffering capacity, and unique amino acid profile. The breastfed infants report higher concentrations of Staphylococci bacteria in comparison to formula-fed infants, more than likely derived from the mother's breast tissue during feeding. Additionally, the beneficial probiotic species Bifidobacterium longum, Streptococcus salivarius, and Lactobacillus gasseri are found significantly more abundant in breastfed infants than formula-fed infants. And when looking at formula-fed infants, formula-fed infants, they exhibit unstable and multifarious bacteria species compared to the breastfed infants. Bifidobacterium is also found dominant, but comparatively lower than the breastfed infants. The formula-fed infants have lower beneficial probiotics comprising their microbiota than breastfed infants, along with higher amounts of bifidobacteria. In analyzing the microbiota of the formula-fed infants, also the lactobacillus content is low, whereas there is significant presence of Escherichia, Enterococcus, and Enterobacter. This finding suggests formula-fed infants have greater diversity of unfavorable bacteria growth while having lower counts of beneficial probiotics. I also looked at the difference between a study formula and a control formula. Infant formula adapted to closer resemble breast milk, meaning high whey, low casein ratio. Low protein, high lactose, and low phosphate concentrations more bifidogenic lead to more. There are no significant differences in infant gut microbiome between the study formula and the study formula plus Bifidobacteria longum, suggesting that the study formula composition can modify gut microbiota by having bifidogenic properties as effective as adding live BL99. So in conclusion, we have seen from what I presented, the difference in nutrient composition between breast milk and formula can have a direct impact on the priming of the gut microbiome in early infancy. Breast milk's composition supports a healthy microbiome by inhibiting the growth of pathogenic microorganisms through nutrient competition and prevention of pathogen adhesion. Thus, breastfed infants reflect a more stable, balanced microbiome dominated by beneficial bacteria, whereas formula-fed infants reflect a multifarious and unstable microbiome with higher counts of pathogenic microbes, along with comparatively lower concentrations of beneficial bacteria. The greater diversity of microbiota in formula-fed infants is dependent on the nutritional composition of the formula. For these reasons, breast milk's bifidogenic properties make it the ideal form of nutrition in the colonization of the infant gut. Therefore, further research is needed to investigate bifidogenic differences dependent on formula composition, allowing for a preparation closer resembling breast milk. My last slide is a link to a research study that Knight's Lab would like to invite moms to participate in that determines the